Moving on, in this part we are going to elaborate the different organelles of the animal cell. So starting from the outer side of the cell, this outer structure, this outer boundary is called cell membrane. The cell membrane of whether a plant cell or whether in an animal cell, we know it very well. In animal cell there is cell wall plus cell membrane. Outer is cell wall, inner is cell membrane. But over here the outermost layer is actually cell membrane in animal cell. So cell membrane is actually a thin semi-permeable membrane. What is semi-permeable membrane? That is, it allows the particles to go inside the cell, and it also allows the particles to go out of the cell. That is, it takes the particles, it ejects the particles. So it is permeable. Permeable is something through which particles or substance can pass through either side. This cell membrane is made up of proteins and fats or lipids, and its function is to protect the cell from external or surrounding environment, and it helps in taking in and taking out of the nutrients. Okay, moving on to the next thing is that this whole cell, animal cell, is made up of a fluid. Okay, this fluid is called cytoplasm. The cytoplasm is actually the fluid within the cell. Why do we need this fluid? Because within the cell there are different organelles that are floating in the cell. They need some medium to move within the cell, to carry out different processes, to carry out cell division, to carry out different digestion processes and different packaging processes. So they need a fluid in order to conduct those processes. So cytoplasm, cyto means cell and plasm means the plasma, that is medium. Now moving on to the next part is nucleus. Let's discuss nucleus. Again, since we know that animal cell is actually an eukaryotic cell, it means that it has a membrane bounded nucleus. We already discussed this. So over here, this is the nucleus of an animal cell, and this two lines or two layers, actually it is a single layer, but I have drawn in a way that you can see that it is actually forming a layer. So this layer is called layer is called nuclear membrane. Nuclear membrane has these small circles that I have shown. These are not just circles, these are pores. So there are pores in the nuclear membrane, and these pores are called nucleopores. So why do we need nucleopores? Because sometimes we need to transfer different components or molecules out of the nucleus or in of the nucleus. So this can happen through pores. So nucleus is porous because it has nucleopores. Now inside the nucleus there is this network of chromatin. It is called chromatin network. It is made up of chromosomes, and chromosome is actually the site where genetic information is present. So chromosomes, or in rough format, when chromosomes are not separated but they are jumbled together in a network form like this, in a mesh form, they are called chromatins, and they contain genetic information, DNA, they contain genes. Okay, within the nucleus there is this, you can see this dark circle over here. This is actually a nucleus. Nucleus is again the center of the nucleus, and overall the nucleus is no doubt the brain of the cell because it carries out major tasks and major processes. So outside this nucleus, you can see that there is this sac-like structure, irregular structure that is originating from the nucleus, or that is actually attached to the nucleus. This is called, as we discussed already in the plant cell, this was present in plant cell as well, it is called endoplasmic reticulum. So there are two types of endoplasmic reticulum: rough endoplasmic reticulum, RER, or smooth endoplasmic reticulum. We discussed the same thing in plants as well. What was the rough endoplasmic reticulum? This area with this dotted region, it is called rough because it contains these dots. These dots are actually ribosomes. So rough endoplasmic reticulum has ribosomes, whereas the outer portion, the outer stacks, have no ribosomes, so it is called smooth endoplasmic reticulum. So ribosomes are present on the rough side and smooth has no ribosomes. What are ribosomes? We discussed already in plants that ribosomes are the material that contain RNA, ribonucleic acid, and they have a purpose to synthesize protein. So since rough endoplasmic reticulum has ribosomes and ribosomes carried out protein synthesis, so RER has this function of protein synthesis, whereas smooth endoplasmic reticulum has no ribosomes but it helps in lipid synthesis. Now moving on to the next organelle, we have mitochondria. Mitochondria is actually known as powerhouse of the cell. This was also present in the plant cell. It is actually an oval shaped organelle that has some stacking inside. These are stacks. Just like points are being stacked over one another. So these are actually the stacked material, mitochondria. It is actually a double membrane bounded. It has two membranes. It is the powerhouse of the cell because it produces energy in the form of ATP, adenosine triphosphate, and it carries out aerobic respiration. What is aerobic respiration? That in the presence of oxygen, we take oxygen and then we burn food to release energy. So it is actually, the name indicates powerhouse. That means it generates power, it generates energy. Energy is generated through aerobic respiration. That is, oxygen is needed in making the energy. Now next to this is this Golgi apparatus, also known as Golgi bodies. These were also present in plant cell. So what do? They are again the same. They are actually flat sac-like structures. They help in manufacturing and their main purpose is packaging. They do packaging, that is whatever the materials are taken inside the cell, food particles, nutrients, whatever it is, they store them. If they are manufacturing something, what they can manufacture, they actually manufacture glycoproteins and glycolipids. So one task is manufacturing. What do? Glycoproteins and glycolipids. Another task is storing and packaging and transporting of the molecules in and out of the cell. So how do they package and store and transport? Through the means of lysosomes. So anything which is transported out of the cell is first comes out of the Golgi apparatus in the form of lysosomes. Lysosomes are actually the cell vesicles. वेसिकल्स मतलब कि छोटे छोटे पार्टिकल्स, छोटे छोटे मॉलिक्यूल्स, छोटा एक मेम्ब्रेन बाउंडेड एक मॉलिक्यूल नहीं कहेंगे, मेम्ब्रेन बाउंडेड ऑर्गेनल होता है, एक स्ट्रक्चर होता है छोटा सा. तो जो भी चीज़ वट एवर द थिंग इज कैरिड आउट ऑफ द सेल, इट ऑलवेज गोज इन द फॉर्म ऑफ लाइजोसोम्स. So lysosomes are membrane bounded, क्योंकि इनको out of the cell move करना है या inside of the cell move करना है, and whatever the nutrient or the molecules they are holding, they should be protected. So that's why they have a membrane. So Golgi apparatus is the site of packaging. पैकेज करता है और फिर उस molecule या nutrient को out of the cell लेके जाता है या inside of the cell लेके जाता है. ठीक है. Then we have this small vacuole. Now this is another contrast between plant and animal. They have smaller vacuoles, whereas we saw in plant cell it was a larger vacuole that almost 90% of the space covered in the animal cell was by a single vacuole. Over here there are multiple vacuoles within the animal cell, because we don't need that much water, because we are not producing energy. We are not actually carrying out, we do produce energy because we need energy, but we do not photosynthesize things. So we need smaller vacuoles which can store food, water, or maybe sometimes if their digestion process happens, not digestion of the human digestive system. I'm not talking about digestion of the human digestive system and the waste that we excrete out. I'm talking about waste product within the cell. Anything which is not desirable in the cell, it is actually on micro level. So they store food, water and waste product of the cell. Cell के अंदर जो भी processes हो रहे हैं, उसमें अगर कोई waste produce हो रहा है या food अगर produce हो रहा है या water या कोई भी nutrient लेके जा रहे हैं, तो वो vacuoles जो हैं वो उनको store करते हैं. And it is double membrane. It is membrane bounded, not double. Okay. So okay, this last structure, as you can see, these are the thread-like structures emerging out of the cell membrane. These are called microvilli. Microvilli are actually the threads that originate from the cell membrane, and they help, since they are thread-like structures, they help in locomotion of the cell. So locomotory action within the cell happens because of the microvilli that originates from the cell membrane. Okay, so one last thing that I can tell you about lysosomes, which were produced by the Golgi apparatus or Golgi bodies, is that again lysosomes are cell vesicles, membrane bounded. They carry out digestion process, digestion of carbohydrates, digestion of proteins, digestion of lipids, digestion of nucleic acid, because they have digestive enzymes. Plus they also carry out the process of excretion, and when the cell is making or breaking, so lysosomes take active part. Okay, one more organelle that I almost forgot to tell you is this. Near the nucleus, this is the nucleus. Near the nucleus are these two small rectangular shaped pair of organelles. These are called centrosomes. What are centrosomes? These are actually the structures made up of tubules. These are actually the tubules, threaded-like structures. They are only functional when cell division happens, whether it's mitosis or meiosis. So what happens during cell division? They become separated. One goes to the north side, another centrosome goes to the south side. These microtubules then become elongated, and what do? They divide the nucleus and the chromosomes or chromatin network into two parts. One part goes to the north side, one part goes to the south side. This happens only during cell division or cell not metabolic, during cell division processes. So usually centrosomes have as not much function in processes of the cell, but it does have in cell division process. That's it for today. Thanks for joining.